Hi everyone, this is the start of chapter 19. In 19.1, we're going to basically be reviewing mutations on gene structure and function. The first part of this will be probably review for most of you, talking about different types of mutations. Also, we'll go into how mutations within non-coding sequences can alter gene function, and also talk a little bit about chromosome structure that can be affected by gene expression. There's a lot of vocabulary in this one, talking about all the different types of mutations. So hopefully there's a couple of different tables that will summarize this, and I'll go over those in a little bit more detail. So gene mutation, a mutation just refers to a heritable change in the genetic material. So some sort of change in the genetic material that can be inherited. This, in the kind of the larger scheme of things, provides allelic variations, right? So we can see variations then besides just sexual reproduction of adding different genes. We can have changes of these alleles. It is the very foundation for evolutionary change, that we have small changes that could be possibly beneficial, although most of them would be detrimental. So they can be detrimental if they result in that allele that functions more poorly than the original. And in general, we think of most mutations as being quite harmful. Organisms have developed ways to repair this, and that'll be kind of at the end of this particular chapter. We'll talk about the different way that they can be repaired. So gene mutations, you want to think about the DNA changes that usually affect one gene. And it can be the change of one nucleotide to another, which is a fairly small one, or deletion or insertion of nucleotides. So some of the ones that you should know about, the first one is called a point mutation, and in this it's just a change in a single base pair. So you can see in the original over here, we would have had a thymine and adenine, and in this case it was swapped out with a guanine and a cytosine. A transition is when the change of a pyrimidine to another pyrimidine or purine to another purine is done. In this case, though, this was a transversion because we actually went from a pyrimidine to a purine. So we went from a T to a G. And the transitions are more common than the transversion. So you would more commonly see a T switch to a C or an A switch to a G, but this one's giving you kind of some idea of what that looks like. Now, hopefully looking at this, you can see that that would just affect one amino acid as we think about what happens through transcription and translation. So let's see, try to get this moving forward. Some other mutations that you should know. A silent mutation means that there is a base change, but the ultimate amino acid remains the same. So think about that with the redundancy and the wobble effect that we saw with that third base within a nucleotide. We can see that often we could have a silent mutation. So even though there is an actual change at the DNA level, the amino acid remains the same. The next one that we can think of is something called a missense mutation, and this is where we have something like we had before, a point mutation, which alters only one amino acid. So when we think of that long polypeptide being changed, one amino acid has changed in that. Sickle cell anemia is one that we see. It's one amino acid substitution, and it actually changes the blood cell morphology, and I think I have a picture of that in a minute. We do see that some of these will not affect the function at all, and it will be what is called a neutral mutation. One of the more detrimental types of mutations is called a nonsense mutation. This is where somewhere along the DNA, a codon was changed. Well, in an mRNA, it would be an mRNA when we get to a codon, that one of the codons has changed to a stop codon early in the sequence. What happens is that this ends up having a very shortened polypeptide. If you have a stop codon early in your mRNA, you can imagine that we would have a much smaller polypeptide. This is just a quick picture showing you of a normal red blood cell, and that we have this one change in an amino acid from a glutamic acid to a valine, and it actually changes the entire morphology of that red blood cell. That tends to be probably less likely that one amino acid could do that, but depending on what it was, what type of a change it was, you can actually have quite a big difference in what would be the morphology of the protein. So mutations can also involve additions or deletions of short sequences of DNAs. Keep in mind that we read DNA, and then subsequently we look at mRNA and tRNA in triplets. If we have deletions or insertions that are multiples different than 3s, what we can imagine is that if we had a deletion here of 4, anything downstream from this would be affected because it's not in that triplet. Same with insertions. If we have an insertion of 4, it means our whole reading frame has shifted. And so anything downstream from this will be changed, every amino acid downstream. We tend to think of these as being, these frame shift as being some of the most detrimental to an organism. So insertions and deletions in that coding region for the proteins can insert those new amino acids, or they can shift that reading frame. Remember I said that the reading frame is actually the more dangerous of this. And frame shift mutations then involve the addition or deletion of nucleotides in multiples of 1 or 2, but not 3. Remember, the 3 would be a codon. It would be either adding or subtracting an amino acid. And so that shifting of that reading frame is actually a huge detrimental mutation. This is just a quick summary, so if you want to kind of review it, this is a good one to look at. So up at the top, we have no mutation, and what we see is we have our different sequences that give us our different amino acids, and so the amino acids are not altered, and likely the effect on a protein would be nothing, right? That's our basic one. A silent mutation, remember, means that we had a base change, but we did not change the amino acid. It remained the same. So no amino acids are altered, which means there is no effect on the protein function. In this next one, we did a base substitution, which then is a missense. Missense just means one particular amino acid has changed. So everything else, if you look up here, has remained the same. It's just changed, substituted one. So you can imagine that if this was maybe at the active site of an enzyme, it might be more detrimental than if it was somewhere else on the enzyme. So in this case, one amino acid is altered, and this can be either neutral. It may not have any effect if it's far away from that active site, or it might be inhibitory, right? It could inhibit that. The next one is a nonsense. Remember, nonsense is where we have a base substitution that changes an amino acid to a stop. And this stop codon means that anything after this will not be translated. If that happens, we would imagine that there are many amino acids that are altered, right? Anything downstream from this. And in most cases, we would imagine that this would be inhibitory. This shortened polypeptide means the protein is not fully formed. And then this last one is the frame shift, which I talked about as being very dangerous. We can have an addition or a deletion of either multiples of 1 or 2 bases. And what we see here is that we get a change. So proline goes to alanine, but everything downstream from this is also changed because our reading frame has changed. In this case, many amino acids have been altered, and we think of this as very inhibitory. So just a little bit of vocabulary to think about. Wild type is what we consider the prevalent genotype. So if we look at a large population, we generally think of wild type as the one that would be most commonly found. Anything that is different from wild type is what we would call a mutant. A forward mutation changes that wild type genotype into some new variation, and that's what we would consider a mutant. We can also, though, with these changes, these forward mutations, can get what we call a reverse mutation or reversion, where that mutant allele actually reverts back to wild type at some point. So neutral mutation, again, this is a lot of vocabulary. Neutral means that it does not alter the protein function. Deleterious mutation lowers the chance of survival and reproduction, so something that would be negative. A beneficial mutation enhances the survival of reproductive success. And so occasionally, a mutation that is either beneficial or deleterious will depend on the environmental conditions. And what we mean by that is that we think of the sickle cell allele as being something that is a negative, right? If you are homozygous for sickle cell allele, you suffer from sickle cell anemia. But what we know, go back one. What we know is that heterozygotes of the sickle cell, they have a slight sickle cell symptoms and kind of a heterozygous of the disease, but they have an increased survival in the presence of malaria, which has allowed this particular, what we would generally think of as a negative mutation, to continue in a population. A conditional mutation is one that affects the phenotype only under specific conditions. So there are some mutants that could be temperature sensitive. This is particularly true in bacterial cells. Sorry. The next thing I want to talk about are suppressor mutations, and these are mutations, a secondary mutation that can affect the phenotypic expression of a first mutation. So we're talking about two mutations in this case. These are different than reversion, even though they may look the same in the sense that maybe it has been given back that function that it did not have, but it's having two different mutations, so it occurs at a second site. We can have ones that are either within the same gene, and these are called intragenic suppressors, so a second mutation in the same gene as the first, or we can have intergenic suppressors, and that means that the second mutation is in a different gene than the first. And when this happens, what we usually see is that a second protein with this new mutation can take on the role of the first protein, or the proteins may act in the same cellular pathway and one may basically make up for the other. In addition, we might see polypeptides that may be subunits, and this second mutation actually helps with the lack from the first mutation. And this is a quick summary of all of those. So you may have this protein here. It gets a mutation. Originally, transport could occur with the first mutation. It is inhibited, but with the second mutation in the same gene, hence the intragenic, it now can function. So that second mutation allows it to continue its job. We can also get this intergenic, meaning it's happening on another gene. So this is our functional enzyme. We get a mutation. It's no longer functional, continues not to be functional. We have another protein that is non-functional, but it now gains a mutation where it can take on the role that this enzyme used to have. And that would be a redundant function. It has the ability to do that. In another case, what we might see is that we have a pathway where one of these enzymes has a higher speed than the other, and the other might be slower. And so they kind of depend on each other to get it efficiently enough that the product is formed. In this case, if this one has a mutation that makes it slow, and this one is also slow, we'd get very little product. What can happen, though, is we have that original mutation. We get a second mutation in the other enzyme in this pathway. And even though this first one is slow, the second one now is fast, and it's made up for the fact that it lost that fast pathway on that first enzyme. We have two more on this one then. We also have what is called a multimeric protein. So we have these two proteins that work together and they're active in this part. If we get a mutation in the first enzyme, the whole entire protein is no longer active. But then maybe we get a compensational mutation in the second part of this protein and now it's active again. This is again compensating for that. And then the last one is looking at a transcription factor. We have this protein that has this normal function. It gets a mutation where it loses function, continues to do that. But now we have this transcription factor, and it can actually turn on a gene that compensates for this particular one. So maybe this pink protein was not being made at all, but once we have this mutation, a mutation in the transcription factor allows this one that can now can come in and compensate for that. I'm going to pause here and then I'm going to finish out this first part on another mini lecture in just a minute.